In a refrigerated storage room in a law enforcement evidence facility, a sealed envelope rests on a metal shelf. The label on its surface was written by hand in ink decades ago. The handwriting is faded. The paper has taken on the particular color that paper acquires after many years in a cool, dry place, and the adhesive along the sealed edge has long since hardened into something brittle and fixed. Inside that envelope, protected by layers of packaging and documented chain of custody forms, is a swab. It was collected from a crime scene on a specific day in a specific location, placed in that envelope by an investigator wearing gloves, sealed, labeled, logged, and stored. What the swab contains, what it has always contained through every year it sat on that shelf, is biological material, cellular material, and within that material, largely intact despite the passage of time and the imperfect conditions of preservation, is a genome, a complete human genetic record, waiting. The waiting is not a metaphor. The envelope has simply sat there, as envelopes do, as evidence does, as the contents of storage rooms do, through the years and then the decades. Investigators have come and gone. The building may have changed. The personnel certainly have. The technology available to the forensic laboratory down the hall has transformed so thoroughly that the instruments now in that laboratory bear almost no resemblance to the ones that existed when the evidence was first collected. The envelope has not changed. What it holds has not changed. And what changed, slowly and then all at once, was what science could do with what it held. The first thing to understand about this process is what traditional forensic DNA analysis was designed to do and what it was not. For decades, the primary forensic DNA method was a technique called short tandem repeat analysis, referred to as STR profiling. The technique works by examining specific locations in the human genome where short sequences of genetic code repeat themselves, and counting how many times each sequence repeats at each location. A person might have 4 repeats at one location and 9 at another. The combination of counts across 13, 15, or 20 such locations produces a numerical profile, a string of numbers essentially, that is statistically unique enough to distinguish one individual from virtually every other person on Earth. That profile can then be entered into a database, and if the same person's DNA appears in that database from a prior collection, an arrest, a conviction, a voluntary submission, a match can be made. The system is precise, it is reliable, it works. What it requires, though, is a direct match. The STR profile from a crime scene must find its identical counterpart somewhere in the database. If the person whose DNA was left at the scene has never submitted a biological sample to that system, has never been arrested, never been convicted, never voluntarily provided material, then the profile sits in the database and returns nothing. Year after year, as new profiles are added to the system, the existing profiles are compared against each new entry, and if the match is not there, the comparison produces silence. The database used by law enforcement agencies in the United States for this purpose is called GODIS, the Combined DNA Index System, maintained by the Federal Bureau of Investigation, and it holds profiles from millions of individuals. For any given crime scene profile, there may be no matching entry in that system. For some profiles, there has never been a match, not in decades of searching. This is where the story of the envelope on the shelf becomes something other than a story about waiting, because what was developing, separately and gradually, in the world of consumer genetics was a completely different kind of database, built on a completely different kind of genetic analysis. The technique is called single nucleotide polymorphism testing, referred to as SNP testing, and where STR analysis looks at a specific and limited number of locations in the genome, SNP testing looks at hundreds of thousands of them, sometimes millions. Across that breadth of examination, the technique captures not just what distinguishes an individual from every other person, but what an individual shares with people who are related to them, people who share a segment of ancestry, who carry overlapping stretches of the same inherited genetic material, who are, in the formal genealogical sense, cousins. The genome is not a monolith. It is a long inherited document assembled from contributions made by parents, grandparents, great-grandparents, and every ancestor before them, each contribution blending and recombining across generations in patterns that follow predictable probabilistic rules. Two people who share a great-great-grandparent will, on average, share a calculable percentage of their genome, smaller than what siblings share, smaller than what first cousins share, but measurable, identifiable. When a SNP profile produced from a crime scene sample is compared against a database of SNP profiles, what the comparison reveals is not just whether an exact match exists, but whether any distant relatives exist in the database, people who share enough of the same inherited genetic material to be identifiable as third cousins, fourth cousins, or close approximations of those relationships, even if they have never met the person whose DNA was collected at the scene and have no knowledge of what their relative may or may not have done. The consumer DNA testing companies that emerged and grew through the 2000s and 2010 CES, companies that offered to analyze a saliva sample and return a report on ancestry and genetic heritage, generated SNP data for their customers by the millions. Their databases became, over time, among the largest repositories of human genetic information ever assembled. People submitted samples to learn about their family origins, to find relatives they had not known about, to connect with distant cousins who had tested through the same service. The data generated for each customer was stored in that company's database and compared against every other profile in that database, producing lists of genetic matches sorted by predicted relationship, second cousin, probable third cousin, possible fourth cousin, and so on down the list. One platform called GED Match occupied a particular position in this landscape. It was not a testing company. It did not collect saliva or produce SNP profiles itself. What GED Match provided was a place where people who had already tested through any company could upload their raw SNP data file, a standardized document produced by the testing process, and have it compared against the raw data files uploaded by every other user of the platform. The appeal was interoperability. A person who had tested through one company could find relatives who had tested through a different company because GED Match accepted files from all of them and compared them against each other, regardless of origin. Genealogy hobbyists, adoptees searching for biological family, people assembling family trees, family history researchers, they uploaded their files and built their matches lists and followed the threads backward into their ancestral lines. What GED Match also became, once the relevance of its database to forensic investigation was understood, was a resource that law enforcement could use under certain conditions. A crime scene SNP profile, a profile generated not from a consumer testing kit, but from biological evidence collected at a crime scene and processed by a forensic laboratory, could, under the right circumstances, be uploaded to GED Match in the same format as a consumer-generated file, and compared against the profiles of everyone who had voluntarily uploaded their data to the platform. Those users had not submitted their genetic information to a law enforcement database. They had submitted it to a genealogy platform for the purpose of finding relatives, but the format of their data and the nature of what it contained made it possible for a crime scene profile to find among them people who shared ancestral segments with the person whose DNA had been collected at the scene. The match list that results from such a comparison looks from the outside like any genealogy match list. It shows percentage figures and predicted relationships. Third cousin, probable third cousin once removed, possible fourth cousin. Each entry is a person identified by whatever display name they chose for their GED match account, carrying a percentage figure indicating how much of their genome overlaps with the profile being compared. What is not immediately visible in that list is what the overlap means in terms of a family tree. Each of those matches is a thread, and the thread, followed carefully, runs backward through generations to a point where two family lines shared a common ancestor, and then, followed carefully forward again from that ancestor, runs toward the person the investigators are looking for. The work of following those threads is genealogical research in its most methodical form. The names on the match list, the third cousins, the probable fourth cousins, are real people with documented lives. They appear in census records, in birth registrations, in marriage certificates, in death records. Their parents appear in earlier census records. Their grandparents appear in records further back still. Working from each match backward to the point of shared ancestry, and then forward again from that ancestral couple through all the lines of descent they generated over the generations, an investigator can begin to map the possible identities of a person whose DNA is in the match list, but whose name is not, because the person whose DNA is in the match list did not upload their data to GED Match. What was uploaded was a profile genetically consistent with them, consistent in the way a sibling's profile is consistent, or a parent's or a child's. The crime scene profile finds the relatives because the profile and the relatives share what relatives share. The process of constructing these family trees is patient and detailed and depends entirely on the quality and completeness of historical records. A genealogical researcher working on an investigative case might begin with a fourth cousin match, a person born in one state with grandparents from another state, whose ancestral line runs back to a couple married in a particular county in the mid-1800s. That couple may have had several children. Each of those children may have had several of their own. And by the time the family tree has been extended across 4 or 5 generations, the researcher is dealing with dozens or hundreds of possible descendants, each of whom carries some probability of being genetically consistent with the crime scene profile. The records narrow the field. Not every descendant of that ancestral couple will have had children. Not every branch of the tree will have produced living individuals. Not every living individual will fit the basic parameters of what is known about the person whose DNA was found at the scene: gender, approximate age, geographic location at the relevant time. The researcher sits at a desk covered in printed documents. There are census pages from the 1900s, from the 1920s, from the 1940s, each one naming the members of a household in a particular place in a particular year, each one a record of who was alive and where they were living at the time the enumeration was conducted. There are marriage certificates with handwritten entries recording the names of the parties, their ages, the date, the location. There are death certificates that establish when and where a line ended, that remove a branch from consideration because it terminated before the relevant period. There are birth records that extend lines forward, that add names to the list of possible candidates, that narrow or widen the pool depending on what they show. The researcher follows each branch methodically, pruning those that end before the relevant generation, extending those that continue into it, noting where records are missing or ambiguous, building a chart that expands across the desk and across the screen, generation by generation, until the field of possible living descendants has been reduced from dozens to a manageable few. The research desk is a quiet place. In this accounting, there is no urgency to the work, or rather, the work does not permit urgency. Methodical genealogical research is not accelerated by urgency. Each document must be found, retrieved, read, interpreted, and connected to the documents that came before it. A census record that lists a family's children by first name only must be linked to a subsequent record that gives full names and birth years. A marriage certificate from one county must be connected to a birth record from another county to establish that the child born in one place is the same individual who later married in another. The connections accumulate slowly. The tree takes shape over weeks and months. When the tree has been extended as far as the records allow, and when the field of possible living descendants has been narrowed by age, geography, and whatever additional information about the person from the crime scene may be available, investigators typically have a small number of candidates. The family tree research has not produced a name in the sense of a certainty. It has produced a set of individuals who are based on what the genealogical record shows and what the genetic match implies. Plausible candidates, people who fall within the right generational range, who were alive and in the relevant area at the relevant time, and who share enough ancestral material with the people on the match list to be consistent with what the crime scene profile would predict. At this point, the investigation shifts. The genealogical phase has done its work. The forensic phase, which began with the original crime scene evidence, now returns to the center. What investigators need to confirm or exclude each candidate is a biological sample, not a sample collected under warrant, not a cheek swab obtained through a formal legal process. Not yet. What they need for the initial confirmatory step is something the candidate has left behind without knowing it was being collected. The biology that makes this possible is the same biology that makes forensic DNA analysis possible in the first place. Every person leaves cellular material behind in the ordinary course of daily life. A paper cup handled and set aside, sipped from and discarded, carries on its rim a small quantity of saliva containing epithelial cells. A straw, a cigarette, a water bottle left in a recycling bin, an envelope licked and placed in a mailbox. These objects are not protected by the legal frameworks governing the collection of biological samples directly from a person. Once discarded, they fall outside the boundaries that would require a warrant. An investigator who observes a candidate discard such an object in a public place may retrieve it, document the retrieval, and submit it to a laboratory for analysis. The laboratory receives the discarded object. A technician in a room lit by fluorescent light, working at a bench where everything is measured and documented, removes what the object carries. A swab is applied to the rim of the cup. The swab is processed. The cellular material yields a genetic profile. That profile is compared against the crime scene profile. The two profiles either match within the statistical tolerances that define a forensic match, or they do not. If they match, the investigation has arrived at its confirmation. The person whose DNA was found at the crime scene has been identified. The name is now attached to the profile. What was an anonymous genetic record has become a person. The paper cup in its evidence bag is not a dramatic object. It does not look like a turning point. It looks like what it is, a paper cup, slightly compressed at the rim where it was held, photographed and sealed in a clear plastic bag with an evidence label attached, sitting on a technician's bench under the fluorescent hum of a laboratory where careful, unhurried work is conducted at all hours. The cup was discarded without any sense that it mattered. It was handled and set down and left behind, as paper cups are, because it had served its immediate purpose and was no longer needed. What it carried on its surface was not intended as a disclosure. It was simply the ordinary biological residue of an ordinary moment. The laboratory technician, pipetting a measured quantity of solution into a processing plate, is performing a step in a sequence. The step before it was documented. The step after it will be documented. The plate will be processed by an instrument that reads the genetic content of the material and produces a data file, and that data file will be analyzed, and the analysis will produce a profile, and the profile will be compared, and the comparison will either confirm or not confirm what the family tree research suggested. The technician's work is exact and careful, and unremarkable in its daily texture. The same measured motions performed with the same precision at each step of the process, the same documentation completed at each stage, the room quiet around the hum of the instrument, the result not yet known. What the STR profile offers, when the confirmatory analysis is complete, is certainty of the kind that forensic DNA matching has always provided, a statistical probability so high that it functions for practical purposes as identification. The numerical profile from the discarded cup matches the numerical profile from the decades-old crime scene evidence at every location examined. The probability that these two profiles belong to different individuals is, in the formal statistical framing that forensic laboratories produce, vanishingly small. The genealogical research identified a candidate. The discarded biological material confirmed what the genealogical research suggested. The two methods have converged on a single individual through two entirely different routes, and they agree. It is useful to hold for a moment what these two routes are and how different they are from each other. The genealogical route began with the crime scene profile, moved outward to find distant relatives in a database, traced those relatives backward through historical records to a common ancestral couple, then traced that couple's descendants forward through generations until the field of living possibilities had narrowed to a workable number. This route operates through probability and inference, not the inference of guesswork, but the inference of careful genealogical reasoning applied to documented records, the same reasoning that genealogists have used for generations to reconstruct family histories from surviving documents. The forensic route began with biological material and compares it directly against other biological material. One route works with inference built on historical records. The other works with direct physical comparison. When they converge, each one strengthens the other. The sealed envelope that sits in cold storage at the beginning of this account was placed there with the expectation that it would eventually find a match in CODIS. That expectation was entirely reasonable at the time. STR profiling was the available method, CODIS was the available database, and the reasonable procedure was to generate a profile, enter it into the system, and wait for a direct match to appear. The match did not appear. The envelope continued to sit on the shelf. The years accumulated. The STR profile was compared against new entries in the database as they were added, and year after year the comparison returned no match, and the case remained what it was, a crime scene profile without a name attached to it. What changed was not the envelope. What changed was not the biological material inside the envelope. What changed was the world around it, the consumer genetics industry that generated millions of SNP profiles and stored them in accessible databases, the genealogy platform that became an interoperable repository for raw genetic data files. The researchers who understood that the same comparison process that helps genealogy hobbyists find distant cousins could be applied, with appropriate modifications, to crime scene profiles. The sealed envelope became newly relevant not because anything about it changed, but because the world developed tools capable of using what it held in a way that had not previously been possible. The laboratory that reprocesses decades-old biological evidence for SNP analysis is working with material that was collected under circumstances very different from the ones in which modern forensic genomics operates. The swab may have been stored in conditions that were adequate by the standards of the time, but that subjected the biological material to some degree of degradation. The sample may be small, not enough for multiple rounds of analysis, requiring careful judgment about how and when to proceed. The DNA extracted from the material may be fragmented, broken into smaller pieces than a freshly collected sample would produce. Modern extraction and sequencing techniques are designed to work with exactly these kinds of imperfect samples to recover enough intact genetic information from degraded material to generate a usable SNP profile, even when the material has been sitting in storage for 20 or 30 or more years. The technician who opens the envelope and removes the swab is doing so with knowledge of what the swab contains and knowledge of what the laboratory's instruments can do with it. The extraction procedure is chosen based on the type of biological material, its estimated age, and the condition of its preservation. Every step is documented. The resulting extract is quantified, how much DNA is present, how intact it appears to be. If the quantity and quality are sufficient, the extract proceeds to sequencing. The sequencing instrument reads the genetic content of the extract and produces a data file containing the profile of hundreds of thousands or millions of individual SNP positions across the genome. That file, in its standardized format, is what can be uploaded to a genealogy comparison platform. The match list that returns from that comparison is the beginning of what the genealogical researcher will spend weeks and months working through. It may contain dozens of entries. Each entry is a real person who uploaded their genetic data to the platform for reasons entirely unrelated to any crime scene. They may have been looking for their grandfather's origins. They may have been trying to find biological family after an adoption. They may have been curious about the percentage of their ancestry attributable to one region versus another. They uploaded a file. The platform compared it against other files, and the file now appears in the match list of a profile that was generated from evidence collected at a crime scene. They do not know this. Their name in the match list carries only their display name and their percentage figure and their predicted relationship: third cousin, probable third cousin, possible fourth cousin. And from those data points, the genealogical researcher begins to work. The genealogical researcher's workspace accumulates over time. What begins as a few printed documents and a mostly blank pedigree chart becomes over weeks a desk dense with paper, census records from multiple decades, marriage registers, birth indexes, death certificates retrieved from state vital records offices and county courthouses, and digitized archives. Notes in the researcher's handwriting tracking which branches have been fully extended, which records are missing, which candidates remain after the branches that have been excluded are set aside. The chart on the screen reflects every addition, each new ancestor added to the tree, each new descendant documented, each new record that either confirms a connection or introduces a complication that must be resolved before the work can continue. The shared ancestral couple at the root of the relevant family line may have lived in the early 1800s. Their children may number 5 or 6 or more. Each of those children may have had several of their own. By the time the tree has been extended forward to the generation alive in the relevant decade, the generation that could have been present at the location where the biological evidence was collected, there may be 30 or 40 or 50 possible living descendants. The work of narrowing that field is the work of records research, finding every descendant's birth, marriage, death, and census record, establishing whether each one was alive and in the relevant location at the relevant time, eliminating those who were not, and retaining those who were. The field narrows, slowly and then more quickly as the constraints accumulate until the remaining candidates are few enough to investigate individually. When the field has been narrowed to a small number of individuals, sometimes 2 or 3, sometimes fewer, investigators move into the confirmatory phase. Each candidate is assessed through available records, and where possible, through discrete observation. The question is not guilt. The question at this stage is biological. Does this person's DNA match the crime scene profile? The genealogical research has produced a set of individuals who are consistent with the genetic evidence. The forensic confirmation will determine which of them, if any, is a direct match. The biological material needed for that confirmation is collected not from the person, but from something the person has left behind. In the ordinary course of a day, in a public place, the paper cup is placed in an evidence bag. The bag is labeled. The label records the date, the time, the location, the identity of the officer who collected it, and the case number to which it belongs. The bag is sealed. The bag is transported to the laboratory, following the same chain of custody procedures that governed the original crime scene evidence. At the laboratory, the cup is processed by a technician who extracts the biological material from its surface, generates an STR profile from that material, and compares the resulting profile against the STR profile on file from the crime scene. The comparison takes time. The documentation of the comparison takes additional time. The results are reviewed by a second analyst before they are reported. The report, when it is produced, states a conclusion. Either the profiles are consistent, meaning they share every measured characteristic within the expected parameters of a true match, or they are not. If the profiles are consistent, the report quantifies the statistical strength of that consistency, the probability, calculated against the relevant population database, that these two profiles could belong to different individuals drawn at random from that population. That probability is reported as a number, often expressed as 1 in many billions or trillions. What that number represents in practical terms is identification. The individual whose saliva was on the rim of the paper cup is, within the bounds of the forensic statistical framework, the same individual whose biological material was recovered from the crime scene, however many years ago. The family tree research pointed toward this person. The forensic comparison confirmed what the family tree research suggested. The sealed envelope on the refrigerated shelf has, through this long chain of steps, yielded a name. The name is now recorded in the case file, alongside the evidence and the documented investigation that preceded it. The envelope itself, still sealed, still carrying its original handwritten label, now faded by decades in storage, remains in its place on the shelf. The biological material inside it has been sampled and processed and returned to storage. The chain of custody documentation records every moment it was handled, every person who handled it, every step it passed through on its way from the original crime scene to the modern laboratory and back into storage again. The case file now contains more than it did before. It contains a profile, It contains a match list from a genealogy database. It contains the pages of a family tree that took weeks to construct. It contains a discarded object in an evidence bag. It contains a laboratory report. The process, traced from beginning to end, moves from a swab in a sealed envelope to a name, and from a name to an arrest, and eventually to whatever legal proceedings follow from that arrest. But the technical arc of it, the path from stored biological evidence to confirmed identity, ends not in a courtroom and not in a dramatic disclosure, but in something considerably quieter. It ends with a paper cup in an evidence bag, sitting on a laboratory bench, in a room where the fluorescent lights hum steadily, and the instruments process their samples without urgency, and a technician produces a report that states, in the measured language of forensic documentation, that the two profiles are consistent. That is the end of the technical process. A cup, a comparison, a conclusion stated in careful, undramatic language, filed in a folder, added to a case record that has existed for longer than most people involved in its resolution can fully hold in mind at once. The whole arc comes down to that. A swab collected decades ago, under conditions that no one at the time knew would eventually matter in the way they came to matter. A database built by people looking for their family histories, who uploaded files they generated for reasons that had nothing to do with any investigation. A researcher sitting at a desk covered in census pages, tracing the descendants of a couple who lived and died a century and a half ago. An investigator watching a man set a cup down and walk away from it. A technician measuring a solution into a plate under fluorescent light. A report, a name. The envelope on the shelf contains all of that, in potential, from the moment it was sealed and labeled and placed in storage on the day the evidence was collected. What it required was time in the slow accumulation of tools and methods and databases that did not yet exist when the ink on its label was still fresh, and the patient work of people who understood that what the envelope held was not a solved case, but a question, still open, still waiting for the science to arrive that could begin to answer it. In a residential neighborhood in East Lampeter Township, Lancaster County, Pennsylvania, on the morning of December 21st, 1992, investigators arrived at a house on Alexander Drive and found what the case record formally documents as a homicide scene. Christy Mirak was 25 years old. She had been teaching second grade at Hans Herr Elementary School, and her students were, at the moment, investigators were walking through that house, waiting for her to arrive at school for what would have been one of the last days before the winter break. She had not arrived. A colleague had come to check on her. What the colleague found is recorded in the case file as the beginning of a record that would remain open and largely static for 26 years. Investigators recovered biological evidence from the scene that day. The evidence was collected, labeled, packaged, and entered into the storage system used by Lancaster County law enforcement, where it was assigned a case number and a place on a shelf alongside other materials from other cases, and it stayed there. The case was active in the years immediately following, and investigators pursued leads and conducted interviews and submitted the DNA profile developed from the biological evidence to the relevant law enforcement databases, and the profile returned no match. It was checked periodically in the years that followed as new profiles were added to those databases, and the answer remained the same, no match. The profile of the unknown male, whose DNA had been recovered from the scene at Alexander Drive, existed in the system as a set of numerical values, and none of the profiles against which it was compared shared enough of those values to produce an identification. the case remained open. What investigators had, from a forensic standpoint, was a short tandem repeat profile, the standard product of traditional DNA analysis, a set of measurements taken at between 13 and 20 specific repeating locations in the genome, converted into a number that could be submitted to CODIS, the National Law Enforcement Database, and compared automatically against the profiles of convicted offenders and other crime scene samples. It was a precise and reliable tool for what it was designed to do, which was to match a known person's DNA against an unknown sample, or to link crime scenes to one another. What it could not do was generate a lead from nothing, could not reach outside the database of already entered profiles and locate a person who had never been sampled. If the man whose DNA was on file in the Mirac case had never been arrested, had never had his DNA entered into CODIS through any of the standard pathways, the profile would continue to return nothing. And so it did, through the 1990s and through the 2000s and into the 2010, accumulating only the quiet negative of no match found. The case file for Christy Mirac continued to exist in that period, as case files do, in a drawer or a box in a filing room, its contents unchanged, the original reports, the evidence logs, the list of leads pursued, the record of the DNA profile, and its successive failures to match. Investigators who rotated onto cold case review would have opened it and read it and looked at what was there, and the question of the unknown male profile would have sat at the center of everything, as it had always sat, waiting for a technology or a database or a circumstance that did not yet exist. That circumstance arrived, eventually, from a direction that the investigators who first worked the case in 1992 could not have anticipated. By 2018, a number of genealogy companies had been operating consumer DNA testing services for several years, and millions of people had submitted saliva samples to receive ancestry breakdowns and family connection reports. One platform in particular, GED Match, had been built to allow users from different testing companies to compare their raw genetic data files in a single place. It was a tool for hobbyist genealogists, and most of the people who uploaded their data to it were interested in finding cousins they had not known about, or filling in gaps in their family histories, or confirming ethnic heritage they had only heard about secondhand. The genetic data they uploaded was not the short tandem repeat profile used in law enforcement. It was a different kind of data entirely, a SNP profile, a snapshot of hundreds of thousands of individual positions across the genome, where single-letter variations occurred. A dataset so much richer in its detail that it could identify biological relationships far more distant than the standard forensic approach could detect. A SNP profile could locate a third cousin. It could find a fourth cousin. From a third or fourth cousin match, a genealogist who knew what to do could begin building a family tree. In 2018, investigators working the Mirac case submitted the crime scene DNA profile to a process of reanalysis, and the result was a SNP-compatible profile that could be uploaded to GED Match and compared against the profiles that users had voluntarily deposited there over the preceding years. C.C. Moore, working with Parabon Nanolabs, conducted the genealogical analysis that followed. The GED match comparison produced a list of matches, not close relatives, not siblings or parents or first cousins, but more distant connections, third cousins and probable third cousins and possible fourth cousins, each one a thread running backward into a shared family history, somewhere several generations in the past. From those threads, Moore began constructing family trees, using birth records, death records, marriage records, census documents, the accumulated paper trail of ordinary lives documented in county courthouses and government archives across the region. She traced the descendants of the common ancestors those distant cousins shared, moving forward through generations, identifying which branches of those families had produced male descendants of an age consistent with the crime, narrowing the candidate pool until the research pointed toward a specific name. That name was Raymond Rowe. He was a local disc jockey known in the Lancaster area who had worked events in the region for years. Investigators confirmed the genealogical lead through a discarded DNA sample, cellular material collected from an object Rowe had thrown away in an ordinary moment, the kind of material a person leaves behind without thinking about it, the kind that carries enough biological information to be typed and compared against a crime scene profile. The comparison confirmed the match. Raymond Rowe was arrested in June 2018, charged with the murder and rape of Christy Myrick, and he subsequently pleaded guilty to first-degree murder and rape. He was sentenced to life in prison without the possibility of parole. The case file that had sat in a filing room in Lancaster County since December 1992 was formally closed, or at least formally resolved. 26 years after the morning investigators first walked through that house on Alexander Drive, it is worth holding for a moment the simple fact of that span of time. 26 years is long enough for the elementary school students who were waiting for Christy Myrick to arrive on the morning of December 21st, 1992, to have grown up, finished school, built careers, had children of their own. It is long enough for the investigative technology that eventually identified Raymond Rowe to have gone from non-existent to commercially widespread to routinely applied in cold case work. The evidence that connected him to the crime did not change in those 26 years. The DNA profile recovered from the scene remained what it had always been. What changed was the context around it, the database, the methodology, the tools available to the genealogist who received the profile and began the careful work of constructing a tree. The Susquehanna River runs along the eastern edge of York County, Pennsylvania, wide and slow in winter, gray against the pale light of a February morning. In February 1988, the body of Anna Jean Kane was recovered from the river in that county. She was 16 years old. The case record documents that she had been sexually assaulted and strangled, and that biological evidence was recovered and preserved at the time of the investigation. Investigators worked the case, interviewed witnesses, pursued leads, and submitted the DNA evidence to the standard databases, and the profile developed from that evidence returned what the Myrick case profile had returned for all those years. Nothing, no match. The case remained open through the late 1980s and through the 1990s and through the years that followed. The evidence preserved, the file maintained, the unknown male profile sitting in storage alongside everything else the investigation had accumulated. Anna John Kane's case did not receive the same degree of public attention over the decades that some cold cases accumulate. It was documented in York County records, periodically reviewed as cold case investigations allow, and carried forward in the system without resolution. The biological evidence collected in 1988 outlasted the original investigation, outlasted the technology available when it was first submitted for analysis, and eventually reached a period in which the genealogical methodology that had produced results in other cases could be applied to it. That application, in Anna Jean Kane's case, led investigators to a man named Scott Grimm. He was subsequently arrested and charged. The details of the genealogical analysis that led to him follow the same general pattern documented in other cases of this kind. the reprocessing of preserved biological material, the SNP profile, the upload to a genealogy platform, the identification of distant relatives, the construction of family trees, the narrowing of the candidate pool, the confirmatory sample. The specific steps are not all a matter of public record in the Kane case, but the outcome is a man was identified, arrested, and charged with the murder of a 16-year-old girl whose case had been open since 1988. The Susquehanna River continues to run through York County, as it always has, and the record it holds, the formal documentation of what began on its bank on a February morning, now contains, after more than 3 decades, the name of the person investigators believe responsible. Fort Worth, Texas, on the night of February 17, 1974, was cold by Texas standards, and the Southwest Bowling Center off Granbury Road was lit and busy for a Valentine's Day dance. Carla Walker was 17 years old. She was at the dance with her boyfriend, and the evening had gone normally, and at some point late in the evening, she was abducted from the parking lot outside the bowling alley. She did not return home. Approximately 3 weeks later, her body was found in a ravine near the town of Peralta, and the case record documents that she had been sexually assaulted. Biological evidence was recovered and preserved. The investigation produced leads, and investigators pursued them, and the case remained unsolved. Carla Walker's case sat in the Tarrant County records for years, then for a decade, then for 2 decades, and then for more than 4. The biological evidence collected when her body was found in 1974 was stored across all of that time, first in whatever format was standard in the 1970s, then in updated storage as preservation practices evolved, carried forward by investigators who understood that the evidence was the center of the case and that the center of the case had to be maintained. What the 1970s could not provide was any mechanism for using that evidence to generate a lead when no suspect was in the database. The profile went into the system, and the system had nothing to match it against, and the decades passed. The investigative turn in the Walker case came through Uthram, a private forensic genomics laboratory based in the Woodlands, Texas, that had built its services specifically around the problem of cold case biological evidence, material that was old, degraded, present in small quantities, or otherwise difficult to work with using standard extraction and sequencing methods. Investigators from the Tarrant County area submitted the evidence from the Walker case to Othram for reprocessing, and Othram's laboratory developed a SNP-compatible genetic profile from the material. It was careful, technical work, conducted in a laboratory with benchtops and sequencing equipment and the methodical attention that old biological samples require, the kind of work that produces no dramatic moment, but simply, after the application of the right methods to the right material over the right amount of time, a usable result. The profile that resulted was compatible with genealogy database searching, and the genealogical analysis that followed produced matches, and from those matches a family tree was built. That tree, worked forward through generations from the common ancestors the matching individuals shared, led investigators to Glenn Samuel McCurley. He was in his 80s. He had been living in the Fort Worth area, the same general region where Carla Walker had been abducted from a bowling alley parking lot nearly 5 decades earlier, and his presence in the Fort Worth area in 1974 was consistent with what the case record required. Investigators sought and obtained a confirmatory DNA sample from McCurley, and the laboratory result confirmed that his profile matched the crime scene evidence recovered in the Walker case. He was arrested in September 2020, 46 years after the night Carla Walker did not come home from a dance, 46 years after the case file was opened in Tarrant County, 46 years in which the biological evidence sat in storage, waiting for a method that could make it speak in a new way. Glenn McCurley was charged with capital murder. The case went to trial in 2021. The prosecution presented the genealogical evidence, the confirmatory DNA match, and the case record stretching back to 1974, and the jury convicted him. He was sentenced to life in prison. The court proceedings in Tarrant County in 2021 were documented in the formal record of the case. Transcripts, filings, the verdict, the sentencing, all of it now appended to a file that had begun as a missing persons report on a February night in 1974 and had accumulated its way through nearly 5 decades of documented investigation toward a resolution that the technology of the 1970s or the 1980s or even the 1990s could not have produced. There is a particular quality to a cold case that has been sitting in a filing system for 40 years. The physical materials in the file have not changed. The photographs taken at the scene are the same photographs. The handwritten notes are the same notes. The evidence logs list the same sealed containers in the same order they were listed when the ink was still fresh. What has changed around those materials is everything else. The investigators who wrote those notes have retired or died. The witnesses who gave those accounts have aged through their lives, and some of them have died too. The neighborhoods and buildings documented in the early reports may have changed or been demolished, and the technology available to the people who periodically reopen the file has undergone a transformation that the people who first compiled it could not have imagined. A biological sample collected in 1974 and sealed in an evidence container is, in one sense, a relic, an object from a different era of forensic science, maintained by institutional habit and the understanding that closing an open case requires more than just setting it aside. In another sense, it turns out it is something else, a resource that becomes more useful as the years pass, not less, because the science available to process it continues to grow in what it can extract from a small and imperfect sample. The 3 cases documented in Pennsylvania and Texas share that quality. The long span of years between the original evidence collection and the eventual genealogical lead, the preservation of biological material through decades of changing technology and changing personnel, the slow accumulation of a method capable of asking the evidence a different kind of question. They are different cases with different victims and different circumstances and different amounts of time elapsed. Christy Mirak's case was open for 26 years before Raymond Rowe was identified. Anna John Kane's case stretched from 1988 to the arrest of Scott Grimm, more than 3 decades. Carla Walker's case ran from 1974 to McCurley's arrest in 2020, 46 years. In each instance, the evidence collected at the beginning of the record was still there at the end of it, still contained in its sealed packaging, still carrying within it the biological information that, read by the right method at the right moment, would eventually produce a name. The genealogical process that produced those names in each case followed a recognizable structure, even if the specific details varied. A crime scene DNA profile, originally developed through traditional short tandem repeat analysis, was reprocessed or supplemented using techniques capable of generating a SNP-compatible dataset. That dataset was uploaded to a genealogy platform where users had deposited their own SNP data voluntarily and for their own purposes. The comparison returned matches, not close relatives, but distant ones, the kind of cousins that a person might not know existed, connected through a shared set of great-grandparents, several generations back. From those matches, a genealogist began the work of constructing family trees, pulling on birth records and marriage records and death certificates and census documents, tracing the descent of each matching individual's family forward through generations, identifying the male descendants of an appropriate age, eliminating those who could be ruled out on geographic or other grounds, and narrowing the candidate pool until only a small number of individuals remained. From that small number, investigators sought biological material from a leading candidate, discarded cellular material collected without the candidate's knowledge, and submitted it for DNA comparison against the original crime scene profile. The match, when it came, was confirmation rather than identification. The genealogy had already found the name, and the confirmatory sample simply established the biological fact that the name was correct. That process, repeated across each of these cases, took different amounts of time at each stage. In some cases, the genealogy work proceeded quickly, producing a strong candidate within weeks. In others, the family tree research stretched over months, complicated by gaps in the historical record, by family lines that were difficult to trace, by the challenge of identifying all the male descendants of a set of common ancestors across multiple generations. The genealogists who conducted this work were researchers as much as scientists, people who spent hours in digital archives and genealogy platforms and digitized census databases, following surnames through counties and states and decades, building a picture of a family spread through time and geography until the picture was clear enough to support a specific conclusion about a specific living person. What the Lancaster County investigators were looking at when they reviewed the Myrick case in 2018 was not simply the same evidence they had always had. It was the same evidence in a new context, a context shaped by the existence of genealogy databases, by the development of techniques capable of generating SNP profiles from crime scene material, and by the emergence of a small community of genealogical researchers who had learned to apply their skills to this specific problem. The evidence had not changed. The context had changed entirely, and the change in context was enough to convert 26 years of no match found into an identification, an arrest, a guilty plea, and a sentence. For the families of victims in cold cases, the experience of a resolution after decades is a matter the public record touches only indirectly. The formal documents, arrest reports, court filings, plea agreements, sentencing records do not capture what it means to receive a phone call after 26 years, or 30, or 46, telling you that the case that had been open since the worst day of your life has produced a result. What the record does show is the fact of the span, the years in the formal record between the date the case was opened and the date of the arrest, documented in the case file the way any other investigative timeline is documented. Those numbers, 26 years, 30 years, 46 years, carry their own weight without requiring elaboration. They are simply what they are, the time that passed. In the Walker case, the time that passed meant that the man who was eventually convicted of capital murder for Carla Walker's death was in his 80s when he stood in a Tarrant County courtroom. The case file beside him bore a date from 1974, the year the record opened, the year the biological evidence was collected and sealed, the year the bowling alley parking lot on Granbury Road became part of a formal investigative record. McCurley had been living his life in the Fort Worth area through all the years the case file sat in storage. He had grown old. The file had not changed. The sentencing in 2021 formally closed what the court record calls an open case, not in the sense that all questions were answered, because cases of this kind often leave questions that no verdict fully resolves. But in the sense that the formal proceeding was completed, the sentence imposed, the record updated with the final entries that a concluded criminal case receives. What had been a number in a filing system since 1974, a case identifier assigned to an unsolved homicide, a set of biological evidence in a sealed container on a shelf, became, in 2021, a record with an end. The case file bearing Carla Walker's name was not destroyed. It was closed in the formal sense, and it continues to exist in the Tarrant County system as it always has, the same documents it has always contained, the same photographs and reports and evidence logs, now with the appended record of an arrest, a trial, a conviction, and a sentence added at the back. 47 years passed between the night Carla Walker did not return from a Valentine's Day dance at a bowling alley in Fort Worth, and the day a Tarrant County court formally closed that record. The bowling alley is no longer there. The neighborhood around it has changed in the ways that neighborhoods change across half a century. What persisted through all of that change was the evidence, the biological material sealed in 1974, maintained through 4.5 decades, submitted to a laboratory in the Woodlands, Texas, and returned as a dataset that led a genealogist backward through family trees to a set of common ancestors, and then forward through generations to a name. The record shows that it worked. The record shows what it found. The record is still there, as it has always been, complete now in the formal sense, with entries at both ends of a span of time that covered most of a human life. On the 26th of June, 1973, in Newport Beach, California, the afternoon was warm in the way Southern California afternoons are warm in June, steady and bright, the kind of day that asks nothing of the people moving through it. Linda O'Keefe was 11 years old. She had been at school that morning, and what the case record shows is that she was last seen walking home along a route she would have taken in the ordinary way, through streets that in the summer of 1973 were, by every account available, simply streets, unremarkable, suburban, sunlit, familiar to her. The drainage ditch where she was found was in Costa Mesa, not far from Newport Beach, a concrete channel beside a road of the kind that runs through many California neighborhoods without drawing notice. The case record opens there, at that location, on that afternoon, with a confirmed finding and a preserved sample of biological evidence that remained in the custody of Orange County investigators for close to 5 decades. The route Linda O'Keefe walked that afternoon has been documented in the case file, the path between the school and the home, the distance, the streets she would have crossed. Investigators worked that route carefully in the summer and fall of 1973, and the case record from those months is substantial. Witnesses were interviewed, physical evidence was collected and cataloged. What the original investigation could not produce, despite the thoroughness documented in those early files, was a match to the biological profile recovered from the scene. The technology did not exist yet to do what would later be done with that evidence. The profile entered into the available databases of the time, and it returned no confirmation, and the case moved into the longer, slower record that cold cases accumulate. Periodic reviews, submissions to new databases as they became available, the file maintained through changes of personnel and changes of method across the decades that followed. Orange County is a large jurisdiction, and the O'Keefe case was not the only unsolved homicide in its files. What distinguished it within the record was its persistence, the continued attention of investigators who did not allow it to be set aside entirely, who resubmitted the biological evidence when new opportunities arose. By the time investigative genetic genealogy had become an established method, The evidence from the 1973 scene was ready to be reprocessed. The original biological material, preserved across nearly 5 decades in the evidence storage system, was submitted for modern extraction and SNP profiling. The resulting genetic dataset was sufficient to work with. The process that followed was methodical: a genealogy database search, the identification of distant relatives, the construction of family trees from birth records and marriage certificates and census entries, the slow narrowing of a branching diagram toward a single name. The Orange County Sheriff's Department announced in 2021 that the research had led investigators to James Allen Neal. The announcement noted that Neal had died before the identification was made. There would be no arrest, no trial, no sentencing. The case was closed, in the formal sense, through identification rather than prosecution. A named individual assigned, at last, to a 48-year-old case number. The record amended to reflect what the genealogy research had found, and then formally closed. What the closure of a case through posthumous identification looks like, in practical terms, is a press release, a case file notation, and a shift in the administrative status of a number that had sat in the open category for decades. For the O'Keefe family, the documentation of that change is part of the public record. For the Orange County Sheriff's Department, it represents the end of an active investigative obligation. What the record contains now is a sequence: a June afternoon in 1973, a route between a school and a house, a drainage ditch in Costa Mesa, biological evidence collected and preserved, a genetic profile extracted and uploaded, a family tree constructed across months of genealogical research, a name and a date of announcement in 2021. The file is no longer open. It is, however, still there. The original reports, the evidence logs, the genealogy findings, the press release, accumulated in layers across nearly 50 years, the whole record available to anyone with access to the official archive. Puget Park in Tacoma, Washington, is a neighborhood park. In March of 1986, it was an ordinary space in a residential part of the city, and on the afternoon of Tuesday, March 26th, Michella Welch was there with her sisters. Michella was 12 years old. The case record documents that she was separated from her sisters at some point during the afternoon, and that she did not return. Her body was found in the park. Biological evidence was recovered from the scene. A male DNA profile was developed from that evidence and entered into law enforcement databases, and the case became one of the files that accumulated across the Pierce County and Washington State investigative record, reviewed periodically. The profile checked against new database entries as they became available. The case never formally closed. The profile sat in those databases for over 30 years. Investigators in the decades that followed the 1986 crime did what investigators do with preserved evidence in an open case. They worked the leads that remained available. They resubmitted to databases. As those databases grew, they kept the file active. What the technology of the 1980s, 90s, and 2000s could not do was match the profile to an individual whose DNA had never been entered into a law enforcement system. The person to whom the profile belonged had not, in the years the profile sat waiting, generated a record in CODIS. That absence is a common feature of cold case profiles. They sit in the database, checked against every new submission, returning no result, year after year, because the relevant individual never intersected with a system that would have captured their DNA. When the investigative genetic genealogy search was conducted in the Welch case, the methodology followed the same documented structure that had been applied in other cases. The crime scene SNP profile was uploaded to a genealogy platform. The resulting matches were third and fourth cousins, distant relatives, people who shared a common ancestor with the unknown individual several generations back, people who had uploaded their own data to the platform for purposes entirely unconnected to any criminal investigation. Genealogical researchers trace those matches backward through the shared ancestral lines and then forward again through generations of births and marriages and deaths, constructing the branches of a family tree from the public and semi-public records that genealogists work with. The tree narrowed. Gary Charles Hartman was identified as the individual to whom the genetic evidence pointed. Hartman was a retired physician. He was living in the Seattle area at the time the investigation reached him. Investigators confirmed the genealogy finding through a separately obtained DNA sample before making contact with him formally. He was arrested in 2008. He was charged with first-degree murder. The Hartman case moved through the Washington state court system in the period that followed his arrest. The charge against him concerned the death of Michelle Welch in 1986, 32 years before his arrest. The case record accumulated a new layer: arrest documents, charging papers, court filings, added to the original investigative record from the March afternoon when investigators first documented what had happened in Puget Park. The park itself has continued to exist. It is still there on the residential street in Tacoma, the location unchanged, carrying in the formal record its designation as the site documented in case files first opened in March of 1986. The record does not require the location to look any different from any other neighborhood park. It simply requires that the location remain part of the documented sequence. The University of Alaska Fairbanks campus sits in a remote northern city, and in April of 1993, it held, as university campuses do, dormitory buildings where students lived during the academic year. Sophie Sergie was 20 years old. She was a Yupik woman, and she was known to people connected to the university at the time of her death. On the 26th of April, 1993, she was found in a bathroom of a dormitory on the campus. The cause of death was confirmed. Biological evidence was recovered from the scene. A male DNA profile was developed from the evidence and entered into available databases. The case became part of the investigative record of the Alaska State Troopers, and it remained there, open, as the years passed. Fairbanks is distant from the forensic laboratory infrastructure concentrated in the continental United States, and the remoteness of the location has been acknowledged in discussions of how the case was handled across the decades that followed. What the record shows is that the evidence was preserved, the case was maintained as an open investigation, and the profile was submitted to databases as those databases developed. For a long time, as with the other cases documented across this record, the submissions returned no result. The person to whom the profile belonged had not, in all the years the case sat open, appeared in a law enforcement database. The investigation remained active in the formal sense, a case number, a file, an obligation, but the evidence it held produced no match. The investigative genetic genealogy search on the Sergie case followed the same structural pattern. The biological material was processed into a SNP-compatible format. The profile was uploaded to a genealogy platform. Matching relatives were identified at the third and fourth cousin range. Family trees were constructed. The research narrowed through the branches of those trees, following the documented lines of descent forward from shared ancestors until a candidate was identified. Stephen Downs had been in the Fairbanks area at the time of Sophie Sergie's death in 1993. By the time the genealogy research concluded, he was living in Maine, on the opposite side of the country. The investigation reached him there. He was arrested in 2019 26 years after the case opened on the University of Alaska Fairbanks campus. The arrest of Stephen Downes in Maine on charges arising from an event in Alaska in 1993 is one of the documented examples of how investigative genetic genealogy reaches across both time and geography. The biological evidence collected in Fairbanks in April of 1993 did not require the suspect to have remained in Alaska. It did not require a proximity that geography might have narrowed. It required only that a distant relative of the person involved had at some point before the investigation uploaded their own genetic data to a platform where it would eventually be compared against a crime scene profile. That upload may have happened years before the search was conducted. The relative who made it would not have known it would one day contribute to an investigation in Alaska. The connection between their personal curiosity about family history and the resolution of a case in Fairbanks is in the documented record, simply the way the method works, a chain of genetic inheritance that runs across generations and across the country, eventually arriving at a name. The case against Stephen Downes moved through the legal system after his arrest. The documentary record accumulated its new layer: arrest, charges, court proceedings, added to the case file, whose earliest documents date from April of 1993. The dormitory on the University of Alaska Fairbanks campus, the building documented in those original records, continues to stand on the campus. The case number that was assigned to the investigation in 1993 continues to exist in the formal record, now with its more recent additions. Sacramento is California's capital, and in October of 1970, it was a city where people lived in apartments and worked and moved through their days in the ordinary way. Nancy Benelighting was 29 years old. She was found stabbed to death in her apartment in Sacramento that October, and investigators responded to that address and collected what evidence was available at the scene and opened a case file that would remain in the active records of Sacramento law enforcement for the next 5 decades. The case is, by the formal reckoning of the record, one of California's oldest homicides to be revisited through investigative genetic genealogy. The biological evidence collected at the scene in 1970 was preserved through all the years that followed, through the shift from paper records to digital ones, through changes in the departments that held it, through the long accumulation of time that separates a 1970 crime scene from a modern laboratory. There is a particular quality to evidence that survives 5 decades in storage. It is not a dramatic quality. The sealed containers holding biological material do not announce what they are or what they have waded through. They sit on shelves in evidence rooms, labeled with the case numbers and dates assigned to them when they were first cataloged, and they continue to be what they are: preserved material, protected from the degradation that would otherwise claim it, waiting for the technology that might eventually do something with what they hold. The Benelak evidence was among the oldest material to be submitted for genealogy analysis in the California cases documented across this period. The laboratory work required to extract a usable SMP profile from 50-year-old biological evidence is more demanding than the equivalent work on more recent material, and the process cannot always be completed successfully. In this case, it was. The resulting profile was sufficient to run a genealogy search. The genealogy analysis of the Benelak crime scene evidence followed the documented methodology. The profile was uploaded to a genealogy platform. Distant relatives were identified among the platform's users. Family trees were constructed from those matches using birth and death records, marriage certificates, census data, and the other documentary sources that genealogical research draws on. The research eventually pointed to Richard Davis as the identified suspect. The case file, which had been open since October of 1970, was amended with that identification. What the formal record now contains is the accumulated documentation of 5 decades of investigation, followed by a genetic genealogy finding, a long, quiet span of open case, and then a name. The apartment building where Nancy Benelak was killed in October of 1970 stands in Sacramento. Buildings endure in ways that cases sometimes do not, and the address in the Benelak case file has persisted in the physical landscape of the city. Through all the years, the case file persisted in the investigative record. A building that was already standing in 1970 continues to stand now, unaware of its designation, in a case file unaffected by what happened within it more than 50 years ago, simply present on the street as it has always been present. The Sacramento address in the Benelak file is in this respect like the Puget Park location in Tacoma, or the Costa Mesa drainage ditch, or the dormitory corridor in Fairbanks, a place that became part of a formal record that carries that designation, whether or not it shows it, that continues to exist in its ordinary way long after the documentation of the events connected to it has grown to fill a considerable file. What runs across these cases when they are held together in view is not a single story, but a common structure. Biological sample collected at a crime scene in a specific decade, a DNA profile developed from that sample and entered into whatever law enforcement database was available at the time. A period of years, sometimes 1 decade, sometimes 2 or 3 or 5, during which the profile was checked against new submissions and returned no result. And then the introduction of a different kind of search, not a direct comparison against a known offender database, but a comparison against a genealogy platform populated by people who had uploaded their own data for personal reasons, many of them unaware that their information could one day become the starting point of an investigation. The genealogy search does not find the person. It finds their cousins, people at the 3rd or 4th degree of relation, connected by a shared ancestor who may have lived a century ago or more. From those cousins, researchers build trees. From those trees, through the patient work of tracing births and marriages and deaths forward through generations, they narrow toward a candidate, and then investigators seek a direct sample, confirm the genetic match, and make contact. In the O'Keefe case, that confirmation process ended at a death notice rather than an arrest. Neil had died before the search concluded, and the case closed by identification rather than prosecution. This is not a failure of the method. The method did what it could do with what was available, which was to name the individual and close the record. In the Hartman case in Washington, the process produced an arrest in 2018 and a legal proceeding that followed. In the Sergie case, it produced an arrest across the full width of the country in Maine and a legal proceeding arising from evidence collected in Alaska. In the Benelak case, it produced an identification from evidence 50 years old. Each case followed its own line forward from the moment of collection to the moment of resolution, and each line was a different length, and each arrived at a different kind of closure. What they share is the archived biological material at one end, and the documented outcome at the other, and the long years of open record connecting the two. Across these cases from the western United States, the biological evidence is the constant. The investigations shifted and returned. Personnel changed. Technology developed. Databases grew. The cases moved in and out of active attention. But the evidence held in the sealed containers in the evidence rooms remained what it was, carrying within it the genetic information that had been present at the crime scenes in 1973, in 1973, in 1986, in 1993, In 1970, the same information in the same molecular form, waiting with no urgency and no awareness for the laboratory method that would eventually know what to do with it. That patience is the kind that requires nothing of anyone. It is not the patience of a family waiting, which has weight and cost. It is simply the patience of preserved material, the organic fact that under the right conditions, biological evidence can outlast decades without losing what makes it useful. The swab in its tube, the slide in its envelope, the sample in its sealed container, these persist. They do not require maintenance in the way an active investigation requires it. They require only the continuation of the basic archival conditions that have kept them intact: the controlled temperature, the sealed environment, the catalog number that connects them to the file they belong to. The California cases, the Washington case, the Alaska case, they are distributed across the western half of the country in places that have little in common geographically, except that they were the sites of crimes in specific decades of the 20th century. Newport Beach is a coastal Southern California city, warm and suburban. Tacoma is a Pacific Northwest port city, gray and cooler. Fairbanks is a subarctic community, remote and cold, in ways that most of the country is not. Sacramento sits in the interior of California, in the broad flat valley between mountain ranges. These are different places with different histories. The cases that connect them to each other do so only through the shared structural fact of biological evidence, preserved and eventually analyzed by the same category of method. The method reached into each of those places and worked with what had been kept there and produced from that work the documented outcomes now held in the formal records of Orange County, Pierce County, the Alaska State Troopers, and the Sacramento District. The time that elapsed between the original crimes and the genealogy-assisted identifications ranges across these cases from roughly 30 to 50 years. Linda O'Keefe's case was 48 years old when the identification of James Neal was announced. Michelle Welch's case was 32 years old when Gary Hartman was arrested. Sophie Sergie's case was 26 years old when Stephen Downs was arrested in Maine. Nancy Benelick's case was 50 years old when the genealogy analysis produced a suspect identification. These are specific numbers attached to specific investigations, and they are worth holding as what they are: documented spans of time during which the cases remained formally open, the evidence remained in storage, and the families of the people named in those files continued to carry the knowledge of their existence. The DNA Doe Project and the commercial laboratories that have worked on cold cases, Paraben Nanolabs and Ultram among them, have processed cases from across the western United States, part of a broader expansion of genealogy-assisted investigation that accelerated after 2018. The cases described here are among the documented outcomes of that expansion, each one a file that had accumulated years of open record before being submitted for a new kind of analysis. The laboratories that received the archived biological material from these cases worked with samples that had been stored for decades, extracting from them the SNP-rich data sets that genealogy platform searches require, and the genealogists who worked with the resulting profiles spent months constructing the family trees that eventually named the individuals the evidence pointed to. The genealogy researchers who worked these cases described the process in terms of patience and iteration. A match list on a genealogy platform does not immediately produce a name. It produces a set of percentage figures and predicted relationship categories: third cousin, possible third cousin, probable fourth cousin, and from those figures, researchers begin the work of identifying who the matches are. tracing their family lines backward to shared ancestors, and then tracing those ancestors' lines forward again through generations to identify all living individuals who could fit the genetic relationship the evidence indicates. The tree grows laterally as much as it grows downward. Every new document consulted opens new branches, new possibilities, new records to check. The narrowing that eventually produces a candidate emerges from the accumulation of eliminations. This branch ends too early. This individual does not match the demographic information available from the crime scene. This descendant lived in a location that makes the connection implausible. What remains after all those eliminations is the person the evidence has pointed to all along. In the Western United States cases, that person was James Neal, deceased. That person was Gary Hartman, a retired physician in the Seattle area. That person was Stephen Downs, living in Maine 26 years after a spring night in Fairbanks. That person was Richard Davis, identified through the oldest biological evidence yet submitted in any of these cases. Each name arrived at the end of a research process that had begun with a distant relative's personal interest in genealogy, a curiosity about ancestry entirely unconnected to crime, expressed by uploading a file to a platform and then forgotten until a researcher's search query surfaced it and began the work of following the connection. The family members of the people whose cases have been described in this record have spoken to journalists and in court proceedings and in the public documents that surround these cases. Their accounts are part of the formal record, and what they describe, the years of waiting, the absence of information, the existence of a case file and a case number and a biological profile that had matched nothing, year after year, is well documented. For the families of Linda O'Keefe, Michelle Welch, Sophie Sergie, and Nancy Benelick, the years the evidence spent in storage were years of a different kind of waiting than the evidence itself experienced. The evidence waited without knowing. The families waited knowing. That asymmetry is part of the record, even when it is not the part the record was built to hold. The cases from the Western United States are not a complete inventory of the genealogy-assisted identifications that have been made in California, Washington, Alaska, and the surrounding states since the method came into wider use. They are a documented portion of a larger set of cases, a portion selected because the facts are confirmed, the outcomes are formally recorded, and the cases represent the range of circumstances in which the method has been applied. Different decades, different locations, different ages of evidence, different outcomes from arrest and conviction to posthumous identification to charges still pending in the legal system. The record of what investigative genetic genealogy has done in the Western United States continues to accumulate, case by case, as laboratories process archived evidence and genealogists follow the trees that result. The Orange County Sheriff's Department press release from 2021, the document that formally announced the identification of James Neal as the suspect in Linda O'Keefe's murder, is a piece of institutional paperwork. It exists in the department's official records and in the public archive. It has a date and a case number and a statement that closes the formal investigative record of a case that began with an evidence collection in June of 1973. The press release is the kind of document that does not announce its own weight. It records a finding in the language of official communication, controlled, factual, administrative. It does not describe what the decades between the crime and the finding looked like from the inside of an investigation or from inside a family. It records the identification and the closure, and that is what formal records do. In some ways, the documents that mark the ends of these cases are the quietest part of the record. The earlier files, the crime scene reports, The witness interviews, the evidence logs from the original investigations, carry more detail, more texture, more of the specific work of trying to understand what happened in a particular place at a particular time. The genealogy analysis reports are technical and precise, dense with the language of genetic percentages and relationship predictions and family tree notations. The arrest warrants and charging documents contain the formal legal language of alleged criminal acts and jurisdictional designations. But the press releases and case closure notices that eventually come, if they come, are brief. They name the person, they state the identification, they note the status of the case. And then the record, which has been growing since the day of the original crime, is formally marked as closed, or as having reached whatever conclusion the facts allowed. The apartment building where Nancy Benelick was found in October of 1970 still stands on its Sacramento street. It is not marked as the site of anything documented in any law enforcement file. The people who pass it or live in it now carry no obligation to know what the case file knows about the address. The building simply continues to be a building on a block in the capital of California, in a city that has changed substantially across the 50 years since a case file first recorded its address. The case file that contains that address has also changed across those 50 years, grown from its original documents into a record that now includes a genealogy finding and a suspect identification. The building knows nothing of this. The file knows everything the investigation was able to learn. The two coexist in their different registers, the physical, standing structure on the Sacramento street, and the formal documentary record in the evidence room, each one a different kind of persistence, each one outlasting the particular years it witnessed, with each one continuing to exist in its quiet way as the record of a case that has now reached the formal boundary of its long open span and come at last to rest. The ferry left Saanich on the morning of November 18th, 1987. It was an ordinary departure, a scheduled crossing, a familiar route, the kind of trip documented afterward in case files as the last confirmed movement of two people before everything else began. Jay Cook was 20 years old. Tanya Van Cuylenborg was 18. They were traveling together from British Columbia to the Seattle area, and the purpose of the trip was practical and unremarkable. Furnace parts, needed for the business operated by Jay's father, a specific order to be collected and brought back. The ferry crossing appears in the case record as the starting coordinate of everything that followed, a point on a timeline from which all subsequent investigation worked backward and forward, trying to account for what happened after the boat reached the other side. What the case file documents after that departure is the progressive accumulation of confirmed absences. Jay and Tanya did not return when expected. The furnace parts were not collected. The people who were waiting for them, who would have been waiting without particular worry at first, then with growing concern, then with the specific and distinct alarm that comes when ordinary explanations have been exhausted, could not account for where the two of them had gone. The case record does not preserve the precise texture of those first hours, but it preserves what followed, the formal documentation of a missing persons report, the beginning of an investigation that would accumulate evidence, sit with that evidence for over 3 decades, and eventually arrive somewhere entirely unforeseen. Tanya Van Cuylenborg's body was found on November 24th, 1987, near Tyee Drive, outside the community of Alger, in Skagit County, Washington. This is where the formal case record begins, in its most detailed form, a location on a rural road in the northwestern part of the state, a place documented in the case file by its coordinates, its proximity to other landmarks, the conditions in which it was found. The investigation found biological evidence at the scene. That evidence was collected, preserved, and processed according to the forensic procedures available at the time, and it yielded a male DNA profile. That profile was entered into the law enforcement database, then maintained for this purpose, where it waited, as such entries do, for a matching submission that did not come. 2 days after Tanya was found, on November 26th, Jay Cook's body was recovered in a rural area near Monroe in Snohomish County. The investigation expanded. 2 counties were now involved. A profile was in the database, and then, by the measure of the calendar, the case went quiet. Quiet is not the same as abandoned. The Snohomish County Sheriff's Office maintained the evidence through the years that followed, through personnel changes, through shifts in forensic technology, through the long intervals when cold cases sit in their storage containers and wait. The people who worked those cases at different points in time are documented in the file, names attached to reviews, requests for additional analysis, the periodic checking of the DNA profile against new database submissions that returned nothing. The profile continued to sit in CODIS, the Combined DNA Index System, the federal law enforcement database into which crime scene profiles and convicted offender profiles are entered and periodically compared. Each comparison that returned no match added one more entry to the record of what had not yet been found. The length of time between a crime and its resolution is not a single experience. It is a series of smaller intervals, each one measured in the actions taken or not taken, the leads that were pursued and then closed, the technological capabilities that were not yet available and then were. Between 1987 and 2018, forensic DNA science underwent changes substantial enough that evidence which had been processed and filed under one set of methods could be reprocessed under another and yield a fundamentally different quantity of information. The biological evidence from the Cook Van Cuylenborg case was among the material that benefited from this. At some point before 2018, the preserved samples were processed using methods that could generate a SNP-compatible profile, a format containing not a numerical code from a small set of markers, but data from hundreds of thousands of individual positions across the genome, a dataset rich enough to be compared against the profiles uploaded by genealogy hobbyists to consumer databases they had joined to learn about their family history. The platform to which the Snohomish County Sheriff's Office uploaded that profile was GED Match. The upload happened in 2018. The person who conducted the genealogical analysis that followed was Barbara Ray Venter, working under contract with the Sheriff's Office. What the database returned when the Cook Van Cuylenborg SNP profile was submitted were not direct matches, no close relatives, no siblings or parents in the system. What appeared instead were distant matches. The case file and subsequent reporting described these as primarily third and fourth cousins, people who shared a small but measurable percentage of their genome with whoever had left biological evidence at the Skagit County scene in November 1987. Each of those matches was a person who had voluntarily uploaded their own genetic data to a genealogy platform, seeking connections to their family history, and who now existed in the database as a thread that could be pulled backward through time. Pulling those threads backward is the work of genealogical research. Birth records, death records, marriage certificates, census documents, local historical records, the accumulated paperwork of ordinary human lives. Barbara Ray Venter's task was to identify the family trees behind those distant matches, trace them back to the common ancestors that those distant matches shared with the unknown person in the evidence profile, and then trace those trees forward again through the generations between the shared ancestor and the present, looking for the living descendants who fit the parameters the evidence established. The unknown person was male. The biology constrained the search in certain ways. The geography of the case, the time period, the available records, all of these informed the shape of the work. The documentation of that work is not fully public, but the structure of it can be followed from what investigators and analysts subsequently described. Months of research, the methodical narrowing of a broad field of distant relatives down toward a smaller and smaller set of possible candidates, until the set became small enough to investigate through conventional means. What the research produced after those months was a name: William Earl Talbot. 2. A truck driver. He was living in the Seatock area, south of Seattle, in the corridor between the two airports. At the time, investigators identified him as a candidate. The evidence profile placed the person responsible somewhere that could be reached from the Skagit County location. The geographic fit was among the factors investigators considered. Before any approach was made, before any arrest, investigators needed a confirmatory sample, biological material from William Talbot to himself, collected outside any confrontational context, that could be tested against the crime scene profile to determine whether the genetic match the genealogy work had predicted was real. The method by which such samples are typically obtained in these circumstances is the collection of a discarded object, something left behind in a public place, something that carries cellular material without any awareness on the part of the person who left it. The case record does not specify the precise form of the discarded material in this instance, but the procedure was followed. A confirmatory sample was obtained. Laboratory analysis was conducted. The result confirmed what the genealogy work had indicated. On May 17, 2018, William Earl Talbot II was arrested. The arrest was made more than 30 years after the ferry left Saanich. You're more than 30 years after the case file was first opened, after the evidence was first collected, after the DNA profile was first entered into a database and returned no match. The arrest did not close the case. It opened the next phase of it, the phase that unfolds in courtrooms rather than in evidence storage rooms and genealogy databases. That phase began formally in May 2019, when the trial of William Talbot II opened in Snohomish County Superior Court, with Judge Linda Crease presiding. The structure of a criminal trial is one of accumulation, evidence presented, testimony offered, the prosecution's account laid out piece by piece for a jury to evaluate. In the Talbot trial, the genealogical methodology was itself a subject of testimony. C.C. Moore, who had not conducted the original genealogy work on this case, but who is among the leading practitioners of the technique, testified for the prosecution. Her testimony explained, to a jury that had not previously encountered investigative genetic genealogy as a forensic method, what it was and how it worked, what a SNP profile contained, how distant matches in a consumer database could be used to construct a family tree, how that family tree could be traced forward through generations to identify a living candidate. The testimony was, in the record of the trial, explanatory rather than purely evidentiary. It was the account of a methodology, a map of the reasoning process, offered so that the jury could evaluate what had led investigators to the defendant's door. The genealogy work was not the only evidence presented. Traditional forensic DNA analysis had been conducted, comparing the crime scene profile to the biological sample obtained from Talbot. That comparison produced what the forensic laboratory certified as a match, a finding expressed in the probabilistic terms that such findings take. The statistical statement of how rarely a random member of the population would produce the same DNA profile. This is the form of evidence juries in such cases are asked to evaluate, not a claim that something is certainly true, but a scientifically derived statement about how probable it would be for the evidence to look the way it does if the person were not involved. The forensic conclusion on the DNA was the most direct connection the prosecution presented between William Talbot, 2, and the biological evidence recovered from the 1987 crime scenes. There was also a physical object. Among the evidence presented at trial was a jacket button, a specific documented artifact recovered near Tanya Van Coilenborg's body in Skagit County in November 1987. The button had been collected at the scene, entered into the evidence record, and preserved for the decades the case remained open. At trial, the prosecution described it as matching a jacket connected to William Talbot, too. The precise nature of that connection, whether the jacket itself was recovered or the match was established through comparison of button type and design, is part of the trial record. What the button represents in the context of the case is something the jury could hold in mind alongside the DNA evidence and the genealogy testimony. A physical object of a kind that could have come from only a limited range of sources, found in a location that placed it as material evidence in the 1987 crimes. Trials involve witnesses for both sides, and the record of the Talbot proceedings includes the defense's responses to the evidence presented. The methodology of investigative genetic genealogy was in 2019 a genuinely new presence in a courtroom, and the defense was not without grounds to question it. The technique had not previously been tested at trial in the way that established forensic methods had been tested over decades. The genealogy work identified a candidate. It did not, by itself, constitute legal proof. The defense's position was that the evidence presented did not meet the standard required for conviction. The jury's role was to evaluate the evidence in its totality. The jury deliberated. On June 28, 2019, the verdict was returned. The jury found William Talbot, 2 guilty on both counts of aggravated first-degree murder, 1 count for the death of Tanya Van Coilenborg, 1 for the death of Jay Cook. The finding was recorded in the case file of Snohomish County Superior Court. It was the first time in United States legal history that a jury had returned a guilty verdict in a case in which investigative genetic genealogy was a central component of the prosecution's evidence. The significance of that precedent did not need to be performed. The case file simply records what it records: the verdict, the date, the 2 counts, the conviction. The fact of the precedent is a matter of legal documentation, a case number in a court system that now carries, alongside the biological evidence and the genealogy research and the jacket button, the formal finding of guilt returned by 12 jurors after deliberation. That finding does not restore what was lost in November 1987. It does not return Jay Cook and Tanya Van Coilenborg to the ferry they boarded from Saanich. It does not change the decades during which the case file held its evidence in storage and waited. What it does is complete a record that had been incomplete for 31 years, adding the final formal entries to a document that began with a missing persons report and a DNA profile and a male suspect whose name was not yet known. The jury's verdict also completed something that extended well beyond this particular case. Before June 2019, investigative genetic genealogy had been used to identify suspects and produce arrests. Guilty pleas had followed some of those arrests. But a guilty plea reached through negotiation between defense and prosecution does not require a jury to evaluate and formally accept forensic evidence. A trial does. The Talbot verdict was the first time a jury had been asked to sit with the genealogy methodology, to hear it explained, to consider it alongside other evidence, to decide whether it contributed to a finding of guilt beyond reasonable doubt and had returned a verdict that incorporated it. That is what the case file means when it is described as a legal precedent, not that it resolved every question about the method, but that it established a documented outcome in which a jury's deliberation and verdict are part of the formal record of what investigative genetic genealogy can produce. The period between November 1987 and June 2019 is 31 years and 7 months, measured in calendar time. In the record of the investigation, it is measured differently in the intervals between forensic reviews, in the years the profile sat in CODIS without a match, in the months Barbara Ray Venter spent constructing family trees from birth certificates and census records, in the weeks between Talbot's arrest and his trial, in the days the jury spent in deliberation. Each of those measurements is a different way of accounting for the same span of time, the same distance between a ferry departure and a verdict. There are things the case file does not resolve. It does not explain, in any document that is publicly accessible, everything that happened between the Saanich Ferry Terminal and the rural roads of Skagit and Snohomish counties in November 1987. The specific sequence of events is not part of the formal public record in the way the forensic evidence and trial proceedings are. The case file holds what it holds: the biological evidence, the genealogy analysis, the traditional DNA comparison, the jacket button, the testimony, the verdict. These are the recorded things. The unrecorded things, the full account of what happened on those roads during those days, exist if they exist anywhere, in forms the case file does not contain. What the case file does hold, in its most complete and current form, is the verdict from June 28, 2019. It holds the 2 counts of aggravated first-degree murder. It holds the name William Earl Talbot, 2. It holds the evidence submissions from a trial that lasted through the early weeks of a June in a courtroom in Snohomish County, where the judge and the jury and the attorneys and the witnesses occupied the same room with the records of a case first opened 31 years earlier. The button recovered near Tye Drive in 1987 was presented as an exhibit. It was cataloged. It was shown to the jury. It was described in testimony. And then it went back into the evidence record, where it continues to reside, a small physical documented object that persists in the formal archive of the case as something that was there at the beginning in November 1987 and was still there at the end in June 29, and that holds within its presence the entire span of time between those 2 moments. The jacket button did not know that it would become evidence. It did not know that it would be photographed and cataloged and preserved for 3 decades, and eventually carried into a courtroom in a county in Washington state to be shown to 12 people asked to decide the facts of a case that began when it was lost on a road in Skagit County. To know nothing. It is an object. It is a round, documented, formally logged object in an evidence collection that now also contains a verdict, and that verdict closes, in the legal sense, in the formal administrative sense, a case that was once only a missing persons report and a question about what had happened to 2 young people who boarded a ferry in British Columbia and did not come back. The trial is over. The verdict is recorded. The case number in Snohomish County Superior Court corresponds to a file that now contains everything the investigation produced: the preserved biological evidence from 1987, the SNP profile developed from that evidence decades later, the GED match database results, the months of genealogical family tree research, the discarded sample that confirmed the candidate's identity, the arrest documentation from May 2018, the forensic DNA comparison, C.C. Moore's explanatory testimony about the genealogy methodology, the prosecution's account of the jacket button, the defense's challenge to the sufficiency of the evidence, and the jury's formal finding returned on a day in late June 2019 that the evidence was sufficient. The ferry from Saanich made its crossing on November 18, 1987. The case file that crossing eventually generated, through the events that followed, through 31 years of investigation and storage and reprocessing and research, is now a complete record in the court system that holds it. Complete in the legal sense, resolved by a jury, formally adjudicated, filed and closed. The questions it does not answer are still the questions it does not answer. The unrecorded things are still unrecorded. The record that exists is the record that exists, and it is, in its current form, as complete as the proceedings of a criminal trial and the findings of a jury can make it. What persists after a trial closes is the evidence, not the proceedings, which are finished, but the physical objects, the exhibits, the samples, the documented artifacts that a case accumulates over its lifetime. The jacket button remains in the Snohomish County evidence system. It is cataloged as a trial exhibit. It is associated with a case number and a verdict date, and the names of 2 people who boarded a ferry in 1987. It sits in a box or an envelope or some form of secure storage in the same county courthouse system that recorded the verdict, in the same administrative apparatus that holds the case file alongside it. It has the specific character of a thing that outlasted the question it helped answer, a thing that was collected in the cold of a November in 1987, preserved through decades in which the question it was part of remained open, and is now part of a closed record in a court that has formally resolved the matter. It was admitted to help decide. The span of time from November 1987 to June 2019 is the span that the jacket button crossed. 31 years and 7 months, give or take the days between specific dates. In that time, the button did not change in any meaningful way. The case around it changed, grew, accumulated, transformed itself from an open investigation to a closed trial. But the button remained what it was when it was found, a small physical object on a road in Skagit County, Washington, now carrying in its formal documentation the weight of everything that happened around it over more than 3 decades. The case record is closed. The evidence remains. The jacket button is still there in the archive of a completed case in Snohomish County Superior Court, a small and quiet and persistent object that carries within its cataloged existence the entire distance between a November night in 1987 and a June verdict in 2019, 31 years and 7 months of an investigation that moved at its own pace from a ferry crossing and an unanswered question to a documented name and a formal finding, settling finally into the still unfinished record of a case that waited and then did not have to wait anymore. In September 2019, the United States Department of Justice released a written policy document, a formal set of interim guidelines, numbered and dated, printed and filed into the official record of the federal government's approach to a method that had, in the preceding 18 months, moved from the margins of forensic practice toward the center of cold case investigation. The document is not long. It runs to several pages. It establishes specific requirements that forensic genetic genealogy may be used by federal law enforcement only in cases involving violent crimes or unidentified human remains, only after other investigative techniques have been exhausted, and only on genealogy platforms that have formally opted into law enforcement access. These were not requirements invented from nothing. The leading practitioners working in the field had by that point already been applying most of them informally as a matter of professional discipline. What the September 2019 document did was take what had been a set of understood practices and place them into the formal, institutional record, give them the structure of policy, assign them the weight of official requirement, and make them something that could be cited, reviewed, and revised. The document exists in a file in Washington, D.C. It is still in effect. It is still the formal framework within which federal agencies operate when they submit biological material from cold case evidence to genealogy databases. That is a particular kind of quiet fact, not dramatic in its content, but grounding in its persistence. A piece of institutional paperwork that arrived at a specific moment in the life of a developing method and said in the language of policy that this is how the method will be governed, at least for now, at least by these agencies, at least under these conditions. The conditions themselves came out of a longer period of uncertainty. Before September 2019, the use of genealogy databases in criminal investigation occupied a space that was neither clearly authorized nor clearly prohibited. The Golden State Killer investigation, whose arrest came in April 2018, had made the method widely visible. It had also prompted immediate and substantial conversation about what the method meant for the people whose DNA profiles sat in those databases, not just the people who had uploaded their own results, but the biological relatives those results implicitly represented. People who had made no choice about any database and who might nonetheless find themselves within the reach of an investigation they knew nothing about. That conversation continued through the months following the 2018 arrests. It was not a conversation that reached a clean conclusion. It was the kind of conversation that produces a policy document and a set of ongoing arguments and a literature of academic and legal analysis and more conversation. The interim guidelines of September 2019 were one response to it. They were not the last word. GDAID, Match, the third-party genealogy platform that had become central to the genealogy-based investigations of 2018, was operating at the time under a default privacy setting that made uploaded profiles available for law enforcement comparisons, unless a user actively chose to opt out. In 2019, following the platform's acquisition by the genomics company Verigen, that setting changed. The new default required users to actively opt in to make a positive, explicit choice to allow their profiles to be included in law enforcement searches. The effect was a substantial reduction in the number of profiles accessible to investigators. The database did not disappear, and law enforcement access did not end. What changed was the size of the pool of profiles that a crime scene DNA upload could be compared against, and the formal basis on which those comparisons were made. Family Tree DNA, another platform, maintained a law enforcement access program under an opt-in framework, operating its own agreement with federal investigators that had itself been the subject of public discussion when it became known in early 2019. The two largest consumer DNA testing companies by database size, 23andMe and Ancestry DNA, had each maintained policies that explicitly prohibited law enforcement from uploading profiles to their systems absent a valid court order. Those policies remained in place. The formal structure of genealogy database access, as it stood in the years following the initial wave of high-profile investigations, was a landscape of different platforms making different choices under different frameworks, with the federal policy guidelines establishing a floor of requirements for the agencies using them, but leaving the architecture of each individual database to the companies that operated those databases. None of this resolved cleanly. The question of which profiles were accessible On which platforms, under which conditions, was not a question with a stable answer. It was a question being answered differently by different institutions and being reconsidered as those institutions responded to public pressure, legal analysis, and their own evolving judgments about what responsible access meant. The evidence rooms were not waiting for those conversations to conclude. In law enforcement agencies across the country, sealed containers bearing case numbers from the 1970s, 1980s, and 1990s had been sitting on shelves through all of it. Through the policy discussions, through the platform changes, through the academic debates, the biological material inside them still viable, still capable of yielding a profile under modern extraction techniques, and still waiting for the combination of laboratory capacity and database access that might eventually convert them from stored evidence into a lead. There were a great many of them. One of the things the emergence of investigative genetic genealogy made plain was the scale of what had been waiting. Cases that had passed through multiple rounds of traditional database searches and returned nothing. Cases where the biological evidence was intact, but where CODIS had simply never found a matching profile, began arriving at laboratories equipped to reprocess them into SMP-compatible formats. The laboratories that had built capacity to do this work found themselves with queues. The cases did not arrive in order of age or urgency. They arrived in the order that agencies submitted them, and agencies submitted them as they obtained funding, established protocols, and worked through the administrative and legal steps required to move evidence from a storage facility to an external laboratory for a new kind of processing. Ethereum, a private forensic genomics laboratory founded in 2018 and based in the Woodlands, Texas, built its business model specifically around processing degraded or low-quantity biological evidence into genealogy-compatible SMP profiles for law enforcement agencies. Cases came to them from agencies that had been carrying open files for decades. Parabon Nanolabs, which had originally built its forensic work around DNA phenotyping, the production of composite facial estimates based on genetic data, added a genetic genealogy division in 2018 when C.C. Moore joined the company. The combination of those two capabilities, a predictive phenotype and a genealogy research program, gave agencies approaching Parabon access to both a visual estimate and a family tree research process operating from the same biological sample. C.C. Moore, by her own account given in media interviews around 2020 and 2021, had personally worked on cases that led to more than 150 identifications within roughly 2 years of the method becoming publicly known. Parabon Nanolabs reported having contributed to more than 200 case solutions by the early 2020s. Those figures describe not a single investigation or a handful of high-profile arrests, but a sustained and continuing body of work, a method being applied, case by case, to the accumulated backlog of cold cases that had arrived at the point where the biological evidence was still there, but the traditional databases had nothing to return. Each of those cases was someone. Each case number was a name or an absence of a name, a family that had waited, a file that had been opened and reopened across years or decades, an investigator who had worked the original case and in some instances was no longer alive to see its resolution. The scale of the work was not visible in any single arrest or any single courtroom proceeding. It was visible in the aggregate, in the cumulative figure that kept rising through the early 2020s as laboratory capacity expanded and database access settled into its new framework and agencies continued to submit evidence they had been storing for a generation. The DNA Doe project operated at one particular point in this landscape. Founded in 2017 by Dr. Margaret Press and Dr. Colleen Fitzpatrick, it was structured as a nonprofit organization working specifically on cases involving unidentified deceased individuals, people found without identification, whose names had not been established through any available means, whose remains had sometimes been held for years in medical examiner offices or interred under case designations that stood in for the names that had not yet been found. The DNA Doe Project assembled a network of trained volunteer genetic genealogists, assigned them to specific unidentified persons cases, and worked through the same process of database comparison and family tree construction that characterized investigative genetic genealogy in its law enforcement application, but directed toward the particular goal of giving an unidentified person their name, rather than identifying a suspect in a criminal case. The volunteer genealogists who worked DNA Doe Project cases did so from home offices and kitchen tables, from desks with genealogy platform windows open and printed census records stacked beside keyboards, working cases the way any skilled genealogist works, a difficult family history, methodically, patiently, following the documented record of births and deaths and marriages and census entries through the generations between a distant database match and the person whose name was still unknown. Dozens of successful identifications had followed from that work by the time the project had been operating for several years. Each identification was its own particular thing, a specific person, a specific set of remains, a specific family that received a specific kind of news after a period of waiting that had in some cases extended for decades. The nonprofit structure of the DNA Doe Project and the commercial structures of Parabon and Othram occupied different positions in the same landscape. They worked from the same underlying science. They used the same databases under the same access frameworks. They produced the same kind of documented output, a family tree narrowed to a candidate confirmed or ruled out, a name placed into a record where a question had been. The institutional forms they took were different, and the cases they worked were different, and the process of getting a case to them was different for a law enforcement agency submitting a homicide file and for a medical examiner's office with an unidentified decedent. But the work itself, at the level of a genealogist sitting with a match list and a blank family tree, was the same kind of careful, patient, document-following work. The consent problem did not resolve because the work was careful and patient. It was a separate question, and it persisted through all of it. Privacy scholars and genetic ethics researchers writing in academic journals and law reviews through the late 2010s and early 2020s returned repeatedly to a particular structural feature of genealogy database searches that no policy framework had yet addressed. A user who opted into law enforcement access on GED Match, or who uploaded their results to Family Tree DNA under its law enforcement agreement, was making a choice about their own data. They were also, by virtue of making that choice, extending the reach of potential law enforcement searches to every biological relative whose DNA overlapped with theirs. Third cousins, second cousins, first cousins once removed, siblings, parents, children, all of whom had made no choice about any database and who might have no awareness that their genetic proximity to a user who had opted in had placed them within the potential reach of an investigation. The argument was not that genealogy database searches were impermissible. The argument was that the opt-in framework, which had been adopted as a solution to the consent problem, did not actually solve the consent problem. It relocated it. The person who opted in had consented. The relatives of that person had not consented to anything and could not consent because the choice that affected them had been made by someone else in a database they might never have heard of. A robust framework for individual consent applied at the level of the person uploading their data did not produce a framework for familial consent because the nature of shared genetic information made individual and familial consent structurally impossible to fully separate. This argument appeared in law review articles. It appeared in academic conferences on bioethics and genetic privacy. It appeared in journalism and in public policy commentary. It was not a fringe position, and it was not a simple one. Researchers who raised it were not, in most cases, arguing that the investigation should stop. They were arguing that the governance framework was incomplete, that the opt-in requirement addressed one dimension of the consent problem while leaving another dimension formally unaddressed, and that the formal record of policy and law had not yet caught up to the structural complexity of what the science made possible. As of the early 2020s, the debate continued. The published arguments continued to accumulate in the formal record. The policy document from September 2019 remained in effect. The platforms maintained their various access frameworks. The laboratories maintained their queues of submitted cases. And the conversations between privacy scholars, genetic ethicists, legal academics, and policy offices continued to produce more arguments, more frameworks, more proposals, without arriving at a resolution that all of those conversations shared. That is what ongoing debates look like when they are recorded in the formal record rather than resolved in it. The record does not close because the question is difficult. The record remains open, accumulating entries as the question continues to be examined from different angles by different disciplines with different priorities and different conclusions. The cases that waited in evidence rooms through all of this were not waiting for the debate to conclude. They were waiting for the combination of circumstances, laboratory capacity, database access, agency resources, investigative attention that would move them from a shelf to a processing queue, to a genealogy researcher's case file, to a confirmed match, to whatever the confirmed match eventually led to. Some of those circumstances were in place, some were not yet in place. Some cases would reach that combination before others. Some might not reach it for years. A very small number might never reach it, not because the biological evidence was not there, but because the particular intersection of resources and timing and access that the method required did not arrive before the evidence degraded, past the threshold that modern extraction could work with. The threshold for modern extraction was not the same as it had been in 1985 or 1995 or even 2005. Laboratory techniques for working with low quantity and degraded biological material had continued to improve. What had been an unusable sample under the extraction methods of one era sometimes became a viable sample under the methods of a subsequent era. That progression did not move in a straight line, and it did not guarantee that every preserved sample would eventually yield a profile, but it meant that the age of the evidence by itself was not a definitive barrier. A case opened in 1971 could produce a usable genetic profile in 2022 if the biological material had been stored correctly and the laboratory applying modern extraction had the techniques to work with what was there. The oldest cold cases to receive genealogy-based investigation in the first years of the method's public practice were from the late 1960s and early 1970s. Each one was its own specific fact, a specific date, a specific location, a specific piece of biological evidence that had been sealed and labeled and stored through decades of technological change, while the investigation that produced it sat waiting in a file. The cases that had been opened in those years were opened before CODIS existed, before PCR amplification was a forensic tool, before the possibility of SNP profiling could have been imagined by anyone working the original investigation. They were opened in a world that had access to none of the tools that eventually moved through them, and they were preserved, sometimes excellently, sometimes imperfectly, through the long interval between the time they were collected and the time when something could be done with them that could not have been done before. The preservation was its own kind of documented effort. Evidence rooms in the 1970s and 1980s were not built around the assumption that biological evidence might be reprocessed and analyzed under techniques that did not yet exist. They were built around the assumption that evidence would be retained in case of retrial or appeal, or in case a known suspect was eventually identified and the physical evidence was needed for confirmation. The idea that an unprocessed biological sample might one day produce a genetic profile capable of being searched against a database of volunteer genealogists was not part of the conceptual framework within which those evidence rooms operated, and yet the evidence was kept. In many cases, it was kept correctly. It survived. When an agency identified a case in its files as a candidate for genetic genealogy processing and submitted the biological evidence to a laboratory, the first step was an assessment, an evaluation of what was present, how well preserved it was, and whether the extraction techniques available could recover a profile of sufficient quality for SNP analysis. Not every submission passed that assessment. Some samples had degraded past the point where modern extraction could recover enough usable material. When that happened, the case returned to the file, the evidence returned to storage, and the investigation remained where it had been, open, holding what it had always held, without the new development that had briefly seemed possible. When a submission did pass the assessment, the extracted biological material was processed through the sequencing equipment that converted it into a SNP dataset, hundreds of thousands to millions of individual data points across the genome, a dataset that could be formatted for comparison against the profiles in a genealogy database. The formatted dataset was then uploaded to whatever platform the investigation was using under the access framework that platform operated, and the comparison produced whatever matches it produced, or, in some cases, produced no matches, or matches too distant to anchor a productive family tree. A case that produced no viable matches was not a failed case in any permanent sense. It was a case whose genealogy lead had not yet appeared. The databases continued to grow as new users uploaded their results. A profile that matched no one in 2020 might match someone in 2023 or 2025, not because the profile had changed, but because the database had grown, and the new entry happened to be from a line of the suspect's family that had not previously had representation in the system. Investigators who had submitted cases and received no matches were told as a standard part of the process that resubmission in subsequent years was worth considering. The database was not static. The landscape of what was searchable continued to expand. It expanded unevenly in the direction of the populations most represented among consumer DNA testing users. The databases were substantially more useful for investigations involving individuals whose families had deep roots in European genealogical lines, because those were the populations most heavily represented in the databases. Cases involving individuals from communities with lower rates of consumer DNA testing participation, including many indigenous communities, many recent immigrant communities, and many lower-income communities who had less access to the consumer testing market, faced a structural disadvantage that the technique itself did not address. A genetic profile was only as useful as the database it was compared against, and the database reflected the choices of the people who had voluntarily contributed to it. That asymmetry was another entry in the ongoing formal record of unresolved questions surrounding the method. It was documented in the academic literature. It was noted by practitioners. It was not resolved by any policy change or technical development as of the early 2020s. It remained part of the honest account of what the method could and could not do, and for whom it worked more readily than for others. C.C. Moore, in interviews and public presentations, described the experience of working a genealogy case as fundamentally similar to what any skilled genealogist does when working a complex family history with incomplete records, following the documented trail of lives backward and forward through generations, holding the ambiguity of imperfect records carefully, returning to primary sources when secondary sources disagreed, and maintaining an ongoing awareness that the family tree being constructed was a hypothesis subject to revision as new information appeared. The criminal investigation context added a layer of procedural requirement, the confirmatory DNA step, the law enforcement chain of custody, the standards that would eventually need to hold up in court, but the core genealogical work was the same kind of patient, methodical, document-following process that family historians had always practiced. That description was useful because it placed the work in the context of an older practice. Investigative genetic genealogy was new in its application to criminal investigation, and new in its reliance on genomic database comparisons that had not existed a generation earlier. But the genealogical reasoning at its center, the construction of family trees from documented records, the narrowing of candidate pools through generational research, the inference of relationships from overlapping genetic data was continuous with a much longer tradition of careful research into who was related to whom and how. The tradition had always been built on the assumption that the records were incomplete and that patience was required. Parish registers missed entries. Census records contained errors. Migration disrupted the paper trail. Family trees built from documented records always carried some degree of uncertainty because the records themselves were imperfect transcriptions of lives as they were actually lived. Investigative genetic genealogy inherited that imperfection alongside the tradition. A family tree built in the process of a criminal investigation was a documented research product, not a certainty, subject to the same kinds of revision and correction that any genealogical research was subject to when new information came to light. The cases where the research converged on a candidate who was subsequently confirmed by a discarded DNA sample and then arrested and charged were the cases that became visible in the public record: the arrests, the trials, the verdicts, the sentences. The cases where the research converged on a candidate who was then ruled out by the confirmatory sample, or where the family tree produced no viable candidate, or where the process was still underway, were less visible. They were the cases still moving through the queue, still in the laboratory, still in the hands of a genealogist working through another layer of census records and marriage certificates, still not yet at the point where anything had been confirmed or announced. The queue was long. It had been long since the method became widely known. It remained long as laboratory capacity expanded, because the number of agencies identifying cold cases as candidates for genealogy processing grew at least as fast as the laboratory's ability to process submissions. The backlog was a documented feature of the landscape, not a failure of the method or the institutions applying it, but a simple consequence of the scale of what was waiting and the time that rigorous processing of each submission required. Time was woven through all of it. The cases themselves were measured in years and decades. The investigations that produced the biological evidence had been conducted in years that were now historical. The detectives who worked those original cases had in many instances retired or died. The victims' families had aged through decades of waiting, decades during which the cases had been periodically reviewed, periodically submitted to new databases, periodically reopened by investigators who believed something more could be done, and then, periodically, closed again when the current tools produced nothing new. The resolutions that genealogy-based investigation eventually produced were not in most cases experienced as events by families who had not been waiting. They were experienced by people who had been carrying the open fact of an unresolved case for 20 or 30 or 40 years, who had in some cases never stopped asking questions, who had in other cases reached a long and difficult accommodation with the probability that the case would not be resolved in their lifetimes. When a resolution came, it arrived into that specific history, into that particular weight of accumulated time, and the experience of receiving it was shaped by all of those years in ways that the formal record of an arrest and a conviction could not fully contain. The formal record was what it was: an arrest, a charging document, a trial, a verdict, a sentence. These were the documented outcomes that the method produced when it produced outcomes, entries in the formal legal record of cases that had previously held only the evidence of a crime and the absence of a named person responsible for it. The formal record did not document the years in between. It documented the beginning, the crime, the investigation, the evidence, and the end when there was an end. The decades in the middle existed in the accumulated weight of a case file that had been opened and reopened across all of that time, not in any single document. Somewhere in a law enforcement evidence facility in the United States, there is a sealed container bearing a case number. The number is written in ink, handwritten or printed, depending on when the container was labeled and how the facility managed its records. The container holds biological material from an investigation that was opened in a year that is now past, a year when the tools that now exist were not yet available, when the database that now holds millions of volunteer profiles did not exist, when the process of converting a biological sample into a genealogy lead had not yet been developed. The material inside the container is preserved. It has been preserved through years of temperature regulation and careful storage, through multiple reviews and submissions and returns, through changes in the personnel who manage the evidence room and changes in the technology available to process what is stored there. The case number on the container corresponds to a file. The file holds a documented record of everything that was known at the time of the original investigation and everything that has been learned or attempted in the years since, the leads that were followed, the database searches that returned nothing, the witnesses who were interviewed and the witnesses who could not be located, the forensic reviews that confirmed what the original analysis had found, and the reviews that introduced new questions. The file is a specific object in a specific drawer or box in a specific record system, as particular in its physical existence as the sealed container on the shelf beside it. The investigation that produced the file and the container is still formally open. It has not been closed. It has not been resolved. The biological material inside the container has not yet been converted into a genealogy lead, or it has been processed, and the database comparison returned no viable matches, or it is somewhere in the queue between submission and result, at a point in the process where the next step has not yet happened. The case sits where it has always sat, in the formal record of an investigation that has accumulated everything it has found and is still holding the question It has always been holding the question of a name that the record does not yet contain. The evidence facility is a building